So my name's Amelia Lane. I'm a postdoc at UCL at the Institute of Ophthalmology, working in Michael Cheatham's group. So I started off doing my PhD in the Department of Biochemical Engineering with Chris Mason and Pete Coffey as my supervisors. And through that I started working on retinal differentiation and came to know Mike Cheatham and his project. We work on disease modelling basically, so it's amazing how we take patient cells, the ability to take patient cells and to study unique genetic mutations in the human context and to make different cell types. So we work on retinal cells, which is photoreceptors, which are something that's been completely inaccessible previously. So our group have worked on a variety of different inherited retinal dystrophies, and we're using the organoid approach, which is where you take iPS, make them into embryoid bodies and then differentiate them in 3 dimensions into a whole retina. We've actually been able to show all the different layers of the retina emerging, so you can really look at the interaction of different cell types. And so we've been looking at retinitis pigmentosa, Leber's congenital amaurosis so far, and a few more patient samples are coming through now, so we're really getting a bank of different disease models. So I think something that's really come up in this conference is trying to get to grips with heterogeneity in stem cell differentiations. So there's some really good approaches going on there in terms of sorting cell types and doing single cell transcriptomics to try to understand what's going on in mixed cultures. So these kind of technologies are really helping us to standardise our protocols and improve reproducibility in what we're doing. We're just starting to get the CRISPR-Cas9 system up and running. So this is a system that really helps us to control for inherent genetic variability, so we can make knockout lines or edit mutations into our control cell lines so that we can have isogenic control. So we have 2 cell lines that behave exactly the same except for the tiny change that makes a protein go missing and might cause retinal degeneration in a dish, which is what we hope to see and treat. So the London Project to Cure Blindness has been sponsored by Pfizer, and it's an initiative to try to transplant embryonic stem cell derived retinal pigment epithelium cells into patients with age-related macular degeneration.